All right, guys, a couple of reminders here. Microscopy lab, okay, is due on Monday, so make sure you're working on that. Okay, getting it finished up. Happy to look it over if you come in. Okay, you can have a look at it. I'll give you some pointers. Also, next week on Monday, we'll be doing another lab, and on Tuesday, an activity. So this week is kind of heavy on the Codere talks and you listen, empty vessel that I must fill kind of teaching, which I hate, which is why I don't like teaching biology. Because biology is a lot of I talk and you have to listen, which I find very boring. Okay, so unfortunately today is probably, definitely, the longest all Codere talks lesson in the whole course. Okay, it's like not fun that way. Okay, but setting up, I will try and give you breaks throughout just so that it's not just me droning at you, okay, the whole class, but there's a lot of stuff to cover. That said, this is also probably one of the most important lessons in this unit because it's where we learn all the functions of the parts of the cell. Okay, so today we're going to be going over all the parts of the cell, what they do. Yesterday we labeled our diagrams. Okay, if you weren't here yesterday, by the way, the diagrams are right over there. Make sure you get one. So we labeled those, but today it's got to be what does each one of those parts do so that next week when you have a quiz, next week on Friday, it's going to be here's a cell, label and give me the function of these structures. Okay? All right, so some big terms today. Okay, key point number one, the significance of compartmentalization. All right, so compartmentalization, okay, is an important term. What it means is cells have different rooms within the cell where different jobs get done. Okay, those different rooms are the different organelles within the cell. Now, something we haven't talked about but will today is that there are actually two groups into which cells can fall. And I don't mean plant and animal, okay? Plant and animal cells are actually in the same group of cells, okay? The two groups of cells, or the two different kinds of organisms, okay, are what we call prokaryotic, and you probably need to add this in somewhere. And eukaryotic. Okay. Those are the two types of cells. Prokaryotic cells are the simplest. Okay? Bacteria are prokaryotes. Prokaryotic cells don't have compartmentalization. So what that means is, if you look at a prokaryotic cell under a microscope, you won't see any of the structures that are on the diagrams we labeled yesterday. Okay? They won't have a nucleus, they won't have a Golgi apparatus, none of that. Okay? A eukaryotic cell does have compartmentalization. So they're generally larger and more complex. Now, yesterday we briefly talked about like natural selection and evolution. If eukaryotic cells are larger and more complex, why are there still prokaryotic cells around if they're like simpler and they don't have organelles? Why would they still be around? Wouldn't natural selection have kind of weeded them out already? Okay. The answer is a little more complex than it might seem. Okay? When you're a eukaryotic organism, yes, you have all these organelles, you have all these rooms where things can happen, and they've got their own environments, and that makes them really efficient. But it also makes you quite a bit more sensitive to changes in your environment. Okay? A prokaryotic cell, because it doesn't have any organelles, can actually endure far more, let's say, inhospitable conditions than a eukaryotic organism. Okay? We find prokaryotic cells, bacteria usually, in places that you or I would fry or freeze. Okay? We found bacteria growing in deep, dark lakes under Antarctic ice. We find them in hydrothermic vents. Okay? We find them in like geysers and hot springs and things like that. No eukaryotic cell would ever survive those conditions, but the prokaryotes do. Okay? Also, conditions that are incredibly like high in minerals or acidic or basic, they survive there. Eukaryotic cells, they can't. Okay? They're too sensitive to those kinds of changes. So the reason prokaryotic cells are still around is they do have certain survival advantages. They can live in places where other types of cells might not thrive. Okay? Now, eukaryotic cells are more complex and generally also more efficient and thus bigger. Okay? If you have all these little compartments, you do things better. Right? So imagine like a prokaryotic cell has to do all of the jobs that an organism has to do, because cells carry out the basic functions of an organism, but it has to do them all in the same room. Okay? A eukaryotic cell has a separate room for every one of those jobs. So imagine this analogy. In your house, you have 3 different temperature environments. Okay? You have the main air environment in which you live. It's about 21 degrees Celsius. You have your refrigerator. It's probably about 4 degrees Celsius. And you have your freezer, minus 15. Right? Are the jobs being done in those 3 temperature environments compatible with each other? Like, if I take the stuff out of the freezer and put it in the air environment that I live in, how long is it going to last? Not long at all. Okay? Similarly, the food in the refrigerator, if I throw it in the freezer, well, then it's frozen and I can't eat it. It's doing a specific job at that temperature. Okay? A eukaryotic cell maintains those 3 different environments. A prokaryotic cell would be like, I don't have a fridge, I don't have a freezer, so I have to keep my house at about 8 degrees Celsius to do all 3 jobs. How well are all 3 jobs getting done at 8 degrees Celsius? Pretty crappy, right? You're cold, your food's gonna spoil, and you can't freeze anything. Okay, so yeah, it's not doing either one or any of them, all 3. Okay, very well. That's the reality of a prokaryote. Okay, it's doing all of the jobs in the same space. Imagine it's storing its DNA in the same place where it digests its food. Is that a good idea? Right? That's not a recipe for bad things happening at all. Okay, accidentally digesting your DNA, that could happen. Okay, so it's not a great situation, but again, they're also more durable in terms of the conditions that they can survive. Okay, so compartmentalization just means you've got those organelles and you can be more efficient because they're doing their jobs in the perfect conditions. Okay, we're going to know the role of each organelle. Okay, we're going to understand cells are a complex system of interdependent processes. Interdependent means they depend on each other. If one part breaks down, they all break down. Okay? Understand how the cell carries out its functions. That's the second point of the cell theory, right? Cells carry out the basic functions. Okay, and understand how materials are transported across the cell membrane, so in or out of the cell, as well as within the cell itself. Okay, so compartmentalization. Eukaryotic cells, okay, and it doesn't say that here, but it should, okay? Eukaryotic cells, okay, are divided into compartments. Every organelle has its own membrane. Some of them even have double membranes, okay? Okay. And so that compartmentalization means that if one process needs it to be a little warmer and maybe have a lower pH than a neighboring process, okay, they can have it. They can have the perfect conditions. And that's, of course, an advantage because if processes are happening in their perfect conditions, they're going to happen better. Okay. So some parts of the cell, for example, might like the temperature in their part of the cell to be a little bit higher. Okay? The mitochondria, for example, tends to run a little hotter than the rest of the organelles in the cell. Okay? That's okay. It's got a double membrane. It insulates the rest of the cell against what's going on in there, and it can have the conditions it wants. If the rest of the cell is running hotter, how well will it run? How do you feel when you have a fever? Pretty bad, right? Well, yeah, you feel cold too, right? Your body's shivering because it's trying to drive your temperature up, okay? But you feel pretty bad when you have a fever. And the reason for that is when your body temperature goes up, a lot of the processes in your body don't work very well. They're designed to run at 37.1 degrees Celsius, not at 38, okay? And so they break down and they don't happen as efficiently and you feel tired, okay? Why does your body do that? Why do you run a fever? Why does your body do that when you get sick? It's partially, it can be a reaction to the viruses, but it can also be a reaction to bacteria. Anything that can live in your body also likes it to be 37.1 degrees Celsius. So if your body runs a higher temperature, those things also don't work as well, and they're easier to kill. Okay? And then you can feel better. That's your body's response. It doesn't always work, obviously. Okay? All right. Everybody okay with that idea? Okay, so the separate compartments are all of those organelles, the Golgi, the nucleus, the rough ER, the smooth ER, all of those are their own separate compartments, and inside of there are the perfect conditions for whatever process is going on. That's compartmentalization. Okay, so the compartmentalization of cells means cells can get bigger. Okay, if things are running more efficiently, cells are naturally going to grow. Now, is there an upper limit to the size the cell can reach? I don't see a cell sitting in this room at a desk by itself, okay? There's kind of a reason for that, okay? Cells sort of have an upper size limit in terms of their ability to transport material. Okay, does it, like, if a cell wanted to, could it get bigger? Yes, but it wouldn't because its ability to transport material wouldn't work as well, and that actually slows it down. Okay, so eukaryotic cells are bigger than prokaryotic cells, yes, okay, but it doesn't mean they can just be as big as they want. Okay, all right. So to give you a size kind of comparison here, a bacteria is a prokaryotic cell. Okay? And through the membrane here, this stuff here that you can see, that's DNA, right? Not safely housed in a nucleus. Just hanging out there, okay, in the cytoplasm with everything else, all right? Where the food's getting digested and the wastes are getting processed, okay? All of that kind of stuff, not a great situation. And you can see that that bacteria, okay, this is half a micrometer. That bacteria might be between 1 and 1.5 micrometers in size. That's really small. Okay? The smallest thing you looked at the other day was about, let's say, 40 to 80 micrometers, okay, the euglena, quite a bit larger by comparison. Okay? An animal cell, okay, is right here. It doesn't even fit in the picture. It's huge by comparison. And a virus is even smaller. Okay? We're certainly not going to see them with our microscopes. Okay? All right. Everybody okay with that idea there? All right, so let's talk about the nucleus and what's in it. Okay, so if we're looking for a kind of a quick way to tell what the nucleus does, okay, the nucleus stores DNA. Okay. And DNA is obviously the code for making materials. If your cell needs to make something, it goes and finds that section of the DNA, makes a copy. Okay, that copy is then used as instructions to build whatever it is your cell needs. is actually a decent kind of representation of what the inside of the nucleus looks like. All these things that look like veins everywhere, that's chromatin, that's DNA. Okay? DNA doesn't always look neat and tidy. Okay? Like sometimes you've maybe seen pictures of chromosomes and they look like X's and V's and things like that. Okay? They only look like that when the cell's getting ready to divide and it's condensed everything and made it all organized. Most of the time, the inside of the nucleus is like somebody took a ball of yarn and completely unrolled the whole thing and threw it in a pile. Okay? It's just everywhere. So imagine being the librarian of a library that has like zero organization. Okay? That's kind of what the nucleus is like. The DNA is just everywhere. Right? It has to go and find the sections that it needs, but that's okay because the nucleolus knows how to do that. Okay? It's like the hoarder that knows where everything in the hoard is. Okay? It's kind of like that. All right, so in the environment of the nucleus, the DNA is stored, it's protected in there, other things don't get in there, it's a nice secure environment. Okay? DNA doesn't get let out. Okay? Copies of it get let out, but the DNA itself doesn't. Okay? And it's got that nice secure membrane around it. Okay? All right, everybody good with that? Now, it does say that from here, protein synthesis is controlled. And the reason it says that is because the nucleolus is in the nucleus. It's actually the nucleolus that does this. Okay, so for this sentence here, this is actually the function of the nucleolus. So on a quiz or a test, okay, I'm not looking generally, like if I've got a diagram of a cell and I say like there's a column that says structure name and then a column that says structure function, I'm not looking for a novel on the function of that particular structure. Okay, like if I said what's the function of the nucleus? Stores DNA. Good. That's all I'm looking for. Okay, I'm not looking for a novel about how chromatin is all unwound and it's all over the nucleus stores DNA. Okay, what does the nucleolus do? It copies DNA or controls protein synthesis. That's it. Okay, it can be as simple as that.
Okay, how would you like look at your DNA? Like, how do people do that if they want to like?
There's actually a number of different ways. If you're just using a light microscope, what you can do is you basically try and catch your cells in the middle of division. Okay? And while they're dividing and all the chromosomes are lined up on the metaphase plate, you can take a karyotype, which basically just means you can take pictures of each of your 46 chromosomes.
Okay.
Okay? If you want to actually look at the DNA itself that makes up your chromosomes, then that has to be like X-ray crystallography. It's a chemical process that will straighten and crystallize the DNA strands. That's what they do for like the Human Genome Project, where they're like, this section of your DNA does this. We know that now, right? After all of that time, we're like, okay, this sequence of base pairs is the gene that codes for your eye color. This sequence of base pairs is the one that codes for your blood type, okay, and so on and so on. Yeah.
Okay.
Okay. Any other questions on that? That's a good question. All right. Now, here's how your nucleolus helps make protein. So we're actually talking about a couple of different structures here. We're talking about the nucleolus, and we're also talking about the ribosomes on this slide. Okay? I'm going to go through this diagram, which is the process of protein synthesis. Know that you do not need to know this process in this level of detail. Okay? All you really need to understand for Science 10 is that the nucleolus makes copies of the DNA, those copies are sent to the ribosomes, and the ribosomes turn them into protein. Okay, they use them to make protein. That's as much as you need to understand protein synthesis at the Science 10 level. The nucleolus copies the DNA, The copied DNA is sent to the ribosomes use it to make protein. That's it. Okay? Now, it's obviously more complex than that, and right away in Bio 20, you'll learn more about this process. So I like to explain and demonstrate the process just so that people get an idea of how much is involved. So I'll give you a minute to get that last bit written down. Nucleolus copies DNA. Copied DNA is sent to the ribosomes. Ribosomes make protein. Okay. Now, in Science 9, you had a short unit on genetics, okay, and you talked about DNA and things like that, okay? And you know that DNA is a double-stranded helix molecule, right? And you probably also know that the sides of that helix pair with each other, okay? A goes with T and C goes with G, right? The base pairs only pair up a certain way, right? And the sequence of those pairs is what makes your genes and things like that. Now, if you understand that, here's how the process of protein synthesis works. If the sides of the strand are complementary, that means if I know this side is A, I know that side is T. Okay, if I know this side is G, I know the other side is C. I only need to make one copy of one side. The other side is a given because A only goes with T and C only goes with G. Okay, so in order to save material and be efficient, what your nucleus or nucleolus does is it uses an enzyme, okay? And again, you don't need to know the name of this enzyme. It's called RNA polymerase. And RNA polymerase pulls the strands of DNA apart from each other, okay? Basically unravels. And then what it does is it copies just one side. It doesn't matter which side because the other side is kind of like the mirror image. Okay, so it copies one side and then the DNA goes back together. What's formed from that is ribonucleic acid. Okay? DNA is deoxyribonucleic acid, two strands. Ribonucleic acid is a single strand. That stuff goes out through the nuclear pores, through those little holes in the nuclear membrane. Okay? That piece of DNA or RNA, sorry, that's copied will then meet up with a ribosome. And what the ribosome does is it clamps down onto it and it starts to read it. And it goes, okay, there's an A, a T, and a G. That means I have to have this amino acid. And it'll go out, get that amino acid, and put it on. Okay? Then it'll read the next three. Okay? That 3 says I need this amino acid. It goes and it chains it on to the next one. That's how a protein is built. Okay? A protein is basically a chain of amino acids. Okay? The ribosomes just make the chain, make the links in the chain and put them together. Okay? It's the DNA that tells it what order to put them in. Okay? And then the protein is formed, and that RNA is just usually reabsorbed. All right? Everybody okay with that? So it's a little bit more complex than what you need to know, but okay, there's a lot of processes that go on to make sure that your DNA stays in here, okay, and only copies of it go out. Okay? But it's the ribosomes that kind of do all the heavy lifting. Okay? All right, so the nucleolus produces RNA, copies your DNA. Okay, a copy of the DNA that contains instructions. That RNA goes outside the nuclear membrane and joins with ribosomes. Okay, and the ribosomes, which actually are also produced by the nucleolus, okay, will make the proteins. They'll assemble the amino acids in the correct order. All right, so what do we need to get from this slide? The nucleolus copies DNA, ribosomes make protein. That's what we need to get from that. Okay. Okay, now, the endoplasmic reticulum. How many different kinds are there? 2. Rough and smooth. Okay, so the rough ER, okay, under an electron microscope actually looks just like this. Okay, so you can see that the layers are kind of folded, okay, over each other, and all the little black dots, all of those are ribosomes. We're not going to be able to see the ribosomes with a light microscope. Okay, they're too small. They weren't discovered until electron microscopes were developed. Okay, but people knew that the rough ER was different than the smooth ER because it looked rougher in texture due to these things that they couldn't really see individually. Okay, so it's covered in ribosomes, looks kind of like this. Okay, the smooth ER, on the other hand, okay, is actually quite hard to see because it doesn't have the ribosomes on it, so it's mostly transparent. Okay, under a special electron microscope, you can just see kind of this end of it here. Anywhere where it actually comes out onto the page is kind of where you can see it. Otherwise, it's just kind of tangled all over and is pretty transparent. Okay, now, for the rough ER, okay, job of the rough ER is to transport proteins. Okay, it's covered in ribosomes. Ribosomes are always making protein, so the job of the rough ER is to transport all of that protein. Now, two things that have kind of a similar name, okay, believe it or not, do very different things. The smooth ER actually has many jobs. Okay? And a lot of them are very closely linked to a cell's survival. Okay? Proteins would very slowly move on their own throughout the cell. Okay? It wouldn't be very efficient, but they would move on their own throughout the cell. But the smooth ER does a ton of things that the cell can't live without. Okay, so if we're talking about the smooth ER, okay, that kind of starts right here. All right, so within the membranes of the smooth ER, here are some of the jobs. Lipids are synthesized. What are lipids? Fat, yeah. Okay, fats are made in the smooth ER. Now, do fats get a bad rap? Now you're saying, oh, fats are bad for you, fats are bad. What's your cell membrane made out of? Fat. Every membrane in your body is a phospholipid bilayer. That means it's made out of 2 layers of fat. Okay? Every cell in your body needs fat. Now, do we as North Americans consume more fat than we require? Yes. Okay? But it's still something that's absolutely necessary for us. Okay? So we consume fats, our body breaks them down chemically, and our cells can put them back together in a form that's useful for building and repairing membranes. That is a super important job that the smooth ER does. It makes these membranes, or these lipids, that get used to build membranes. Your membranes are always getting damaged and things like that. They're in constant need of repair. So that is a super important job. Okay? Lipids are synthesized for building or repairing membranes. Okay? Another thing that goes on, because the enzymes for doing this are similar, okay, is that complex carbohydrates can be broken down. That would be starches. Okay, so if you went out and you had a big rice bowl, okay, that's a lot of starch. Or you had, a couple of baked potatoes. There's a bunch of starch there. A big plate of pasta. Okay, you consume all these complex carbohydrates, which are basically just chains of sugars. The smooth ER helps to break those into smaller sugars so that your lysosomes and other organelles can then use them for fuel. Okay, so it does break down complex carbohydrates. Okay? Generally tries to turn them into things that are more useful. Okay, so complex carbohydrates get broken down, and drugs and poisons can be detoxified. Not all of them, obviously, but okay, the ones our body is capable of breaking down, okay, at a cellular level, the smooth ER helps to break them down. It's important that that happens inside the smooth ER, because usually the byproducts of that are nasty. Okay, so if that happens inside the smooth ER, it can only damage the smooth ER, whose job is to make lipids to repair itself. Okay, so it's good that it's kind of being done in there. Okay, the smooth ER has also got lots of access to the cell membrane, so anything really bad can get, sent out pretty quickly. Okay, so what organ in your body has the same job, detoxifying things? Your liver. Yeah. Okay. If you look at your liver cells under a microscope, they are filled with smooth ER. Okay? They're specialized because that's their job. Their job is to detoxify poisons. So the cells in the liver have way more smooth ER than a muscle cell. okay? Because their job is to help break all that stuff down, okay? So your liver does that on kind of a larger scale, okay? Not that you guys would have had ever any experience with this because you're very young, okay, too young, but that's kind of what a hangover is, okay? So if you've overindulged, we'll say, okay, the next day you feel awful, right? You are nauseous and you have a headache. The headache's usually the result of dehydration, okay? But the nausea part is actually the result of your liver breaking down the alcohol, okay, into other less toxic forms, okay, which then go into your small intestine. That's where the nausea comes from. That stuff is very irritating. Okay? The byproducts of breaking down a toxin like alcohol, okay, is going to make you feel rotten. Okay? Anytime you're sick, that's what's going on. If you're feeling nauseous, there's something irritating your small intestine. Okay? All right. Everybody with me there? Okay, so lots of important jobs. Okay, last one. Fat-soluble hormones. So not protein-based hormones, but fat-soluble hormones can also get produced here. On a test, I do not require you to know all 5 jobs. Okay? You can give me 1, I'm good. All right? Now, does that mean you should still know what the others are? Yes. Okay, there could be a multiple choice question, then you would have to know which ones would apply. But if I was asking you to write it out, 1 would be good. Okay, and as I was saying before, the reason all of these tasks are performed here is because often the byproducts of these are things that need to get out, okay, or we don't want to have in contact with more sensitive parts of the cell, okay, so it's good to have them go on inside the smooth ER. All right, so rough ER transports protein. Smooth ER, okay, it detoxifies poisons, breaks down complex carbohydrates, makes lipids, builds fat-soluble hormones, okay, any of those is fine. Okay. Anybody need that one anymore? All right, so this is just more about the lipids here. Lipids then can get moved quickly to the cell membrane. That's usually where they're needed. Okay, if we're looking at other representations of this, this actually might be a little bit better electron microscope picture. Here's the rough ER on the right. This is the smooth ER here on the left. Okay, so you can see that this is just all kind of, it's like you took a whole bunch of cooked spaghetti and just threw it in a pile. That's kind of what the smooth ER, okay, would look like. It's just a pile of stuff all intertwined. Okay, but the rough ER is much more layered and organized. All right, Golgi apparatus. Okay, yesterday I said the Golgi apparatus, if you wanted an analogy, is kind of like the Amazon sorting facility, okay, that's in Balzac, okay, up by the big mall there. All right, it's actually like just a little bit southeast of the mall. So what goes on there is all the companies that sell stuff to Amazon ship their stuff to that place. Okay, that's what Amazon would call receiving. They're receiving the goods from their vendors. Okay, once that stuff is there, okay, then the people that work in that building have to go, okay, so-and-so ordered this, so we need to make sure that we package that order and make sure that it gets on this truck because this truck is going to that neighborhood and it can make the delivery. Okay, so it's receiving, getting it from the vendors, and then shipping, sending it out on a truck to go to your house and end up on your doorstep when you get home from school today. Okay, that's what the Golgi apparatus does. Stuff comes into it from the cell that requires a little processing, okay, and some packaging, more than likely, and then that package gets shipped to the cell membrane. Okay, no, it doesn't use a truck, it just uses this envelope that's basically made of the same material as a cell membrane. All right, so we can kind of see that here. Okay, so the Golgi apparatus actually has 2 sides. Okay, it has the sys space, you don't need to know that. Okay, that's the receiving side, stuff comes in on that side, and then it's packaged. So you can see this little thing here, okay, is going through the the Golgi apparatus, and when it comes out of the Golgi apparatus, it's actually in what we call a transport or secretory vesicle. It's basically put in an envelope that's made of the same material as the cell membrane, and then when that touches the membrane, the envelope just becomes part of the membrane, and what's inside of it just pops out. Okay? So it would be like, this is going to sound a little bit gross, but if you've ever like picked up after your dog, right? You have the little bag that you put over your hand and you pick up their leavings. Okay, so it would be like you've got, you know, the bag is over your hand, you enclose it and you turn it inside out, right? So that you never actually have to touch it, it's inside that vesicle, okay? It would be like when it gets to the cell membrane, it would be like turning it back out again and popping out what's inside, okay? That's kind of what happens here. It comes in one side, it gets packaged. When that package gets to the membrane, it just pops out what's inside of it, okay? Because, well, cells don't care. They're just getting rid of it. Maybe they need to ship it out. Maybe it's just something they want to get rid of, okay? But that's how they do it. All right, so the Golgi apparatus has a cis face, that's the incoming or receiving side, the trans face, and you can see that on the two faces look a lot different. Okay, this side's quite smooth because things are just coming in on that side, but this side's like all bubbly. Okay, well, that's because every one of these bubbles that comes off has got something in it that's being shipped out. Okay, everybody all right with that? Okay, so Golgi apparatus basically just, if we're looking for a quick way to say it, it packages materials for removal from the cell. Okay, packages materials to be removed from the cell. Okay, under an electron microscope, this is what it looks like. This is the cis face here on the bottom, and this is the trans face here. And you can see all these bubbles, okay, all these transport vesicles coming off of that shipping side, okay, and heading out. Okay, I'm going to give you guys a little 5-minute break here, okay, because I've been talking steady here. So I'll give you a little 5-minute break to reset, and then we'll pick up here. Okay. So, next thing we got to talk about, lysosomes. They're only going to be in what kind of cell? Animal, right. Lysosomes digest your food, okay? So that is their main job, okay? If you want to have like a quick function for what the lysosomes are, lysosomes digest food. Okay, and they're only going to be present in animal cells because obviously plants don't eat food. Now, lysosomes do have to have a very special membrane, okay, because what's inside of them is digestive enzymes. And those digestive enzymes are designed to digest food. Well, as a chemoheterotroph, again, word from yesterday that's important, okay, what do you eat? You eat other animals and plants, other organisms, right? And what are those things made out of? Cells. So the stuff in there is designed to eat cells. Do you want that getting out? Because it will eat your own cells, okay? It's pretty harmful stuff. So you want it kept inside of these special membranes that are very strong and resistive to that. In the same way that inside your stomach, okay, you have a special mucus lining that's secreted by the inside of the stomach that protects it from the stomach acid and enzymes that are inside your stomach. Okay? If that layer breaks down, okay, lots of different things can cause that, you can get a what? Starts with a U. You guys ever heard of an ulcer? Okay? An ulcer is when the digestive enzymes and acid inside your stomach actually chemically burn the inside of your stomach. Okay? Usually just means that the mucus lining has gotten thin. Lots of things can cause that. Okay? Long-term stress and anxiety can lead to poor production of mucus on the inside. Alcoholism, that dissolves the mucus lining as well. Okay? Things like that. There's lots of things that can cause that lining to get compromised. Okay? How many people have ever had heartburn? Okay? Heartburn is what an ulcer is kind of like. It just means that some of the acid inside your stomach got past the little muscle on the top of your stomach and got into your esophagus. And it burns the inside of your esophagus because it doesn't have that mucus protection. Okay, so if you got acid reflux, some of the acid comes out of your stomach and gets into the esophagus, okay, and burns. Same if you vomit, okay, you feel that burning sensation, it's the same thing.
Ashley was wondering, how do you know if you have heartburn?
If you have heartburn, you'll basically just feel like a bit of a warming kind of pain right kind of here, just above your heart. Yeah, that's where your esophagus is. Your stomach is actually right about here. Yeah, and so you feel it in your esophagus. Your stomach's right about here. Okay. Right where your rib cage kind of comes together, okay? Yeah, it's like right here. I had a little high. The heartburn would be right about here.
So where's
Okay, your stomach is right about here. Yeah, right? You feel where your rib cage kind of comes up in the middle, and your stomach is kind of right there. We call this our stomach, but your stomach is not there. Okay? Your intestines are there. That's where your bowels are. Okay? Your small intestine, your large intestine, that's all in there. Your liver goes across like this. There's a whole bunch of organs all crammed into one spot there, right behind your rib cage, because your rib cage protects them all. Right? That's what it's for. Okay, now, your lysosomes don't just digest food. They'll digest anything. Sometimes organelles within the cell stop functioning. They just break down for whatever reason. Okay? Well, your cell is the ultimate recycler. Okay? If it sees a mitochondria, for example, that's no longer working, it'll just treat it like it's food. Okay? It'll eat it, recycle it, okay? Turn it into something else. Okay? Nothing wrong with that. The other function of the lysosome in animal cells is as a self-destruct mechanism. Okay? Believe it or not, your cells have a self-destruct mechanism. Okay? It is designed to keep your cells from becoming malignant. Okay? So if your cell becomes damaged, especially the DNA, it can start to do things that are harmful. One of the biggest things is divide out of control. What do we call that? Cells that are dividing out of control. That's cancer. Okay? Cancer is caused by your cells dividing rapidly when they're not supposed to. Okay? That usually forms a tumor, a lump of cells that are malfunctioning. Okay? The thing about cancer that makes it hard to treat is that your body doesn't really see anything wrong with it. Those are your cells. They're supposed to be there. But when they're growing out of control like that, they'll form a larger and larger tumor that consumes more and more resources from the healthy cells around it, destroying the healthy tissue. So if you got a tumor on your liver, for example, it would destroy a lot of the healthy liver tissue, and that can obviously be bad.
How do the cells grow out of control?
One of the first things that can be damaged is the mechanism that controls cell division. So if that gets damaged, and it's part of your DNA, okay, if it gets damaged, the cell just divides and divides and divides and divides, and that's what leads to that overproduction of cells that form a tumor. Okay, that can be caused by lots of different things. Lots of things are carcinogens, right? There's chemicals that are carcinogens. Ultraviolet light can cause skin cancer. That's the same thing. It's rapidly dividing skin cells in one spot. Okay, lots of things can cause that to happen. Now, most of the time, your cell can detect if it's malfunctioning. And if it is, it just ruptures all the lysosomes inside. Okay? And the cell will eat itself to death. Okay? But it's taken one for the team. Okay? It's like, I know that something's wrong and I'm going to be harmful to everybody else, so it'll destroy itself to preserve the rest of the cells around it. Now, unfortunately, one of the first mechanisms to go when a cell becomes malignant is that exact mechanism, and then it doesn't detect there's something wrong and starts to divide out of control. But it does work some of the time. Okay, so inside a lysosome, okay, there's not much to them. They're just a bubble, okay, bubble with a special membrane, and there are hydrolytic enzymes inside that do all the digestion. Okay, so lysosome, digest food. Okay, all we would need. Okay, mitochondria. So the mitochondria, this is in both. Plants and animal cells have a mitochondria. This is where cellular respiration occurs. So if you wanted a quick function of the mitochondria, cellular respiration. Okay. Sugar is broken down in the presence of oxygen, producing carbon dioxide and water. Okay, so the mitochondria actually has a double membrane. Okay, that double membrane actually acts as a bit of an insulator against the reactions that go on in here. It is kind of a combustion reaction, and combustion reactions produce a lot of energy, even thermal energy. Okay, so if you're working out or you got a fever, these are the organelles that are working overtime. Okay, they're consuming more energy, they're producing more heat, okay, and you get warmer. Okay. All right, so under an electron microscope, okay, they look pretty much like the drawing. Okay, you can see the folds in there. What's the purpose? Lots of the organelles we've looked at have folds. What's the purpose of the folds? Okay, let me ask you this. Which has more area? This or this? Okay? Which has more surface area internally, like inside? This one or this one? Actually, it's the one on the right. It's counterintuitive. It seems like, oh, well, you filled this up, it has less. No, I filled it up with more surface area. In this circle, all the surface area is on the inside of the membrane. But if that membrane folds over itself, I don't just have the interior of the membrane, I've also got all of the folds as surface area as well. Your intestines look like this. Okay? They fold over themselves in something called villi that helps you to have a greater surface area for absorbing the stuff that you digest in your food. Okay? In fact, if you were to lay out the entire surface area in your small intestine, it would cover a tennis court. Yeah, crazy. Okay? So anytime you see something that has folds in it like that, like the rough ER had all those folds in it as well, okay, it's just trying to increase the surface area that's available for whatever job is being done there. Having those folds inside the mitochondria makes a greater surface area for cellular respiration to occur. Okay? All right. Questions on that one? All right. Chloroplasts. Only going to find those in? What's that mode of nutrition? Okay, that's the process. What's the mode of nutrition called? Photoautotrophs, okay? So photoautotrophs are going to have chloroplasts, okay? And if you want the function of the chloroplast, that would be it. Chloroplast, photosynthesis, okay? That would be all you would have to write. Okay, chloroplasts also have a double membrane. Okay? Absorbing solar energy means that you're also absorbing a fair amount of heat. Okay? So they do need to insulate against the rest of the cell. Now, when we do our cell anatomy lab on Monday of next week, okay, we're going to look at the cells in the kind of upper layers of a leaf. And they're going to look pretty much like this diagram right here. And you are going to see fairly large grayish green oval structures. You're going to see the chloroplasts. Okay? They'll be very plentiful, very large and obvious, okay, for you to see. All right? Typically, inside of a chloroplast, okay, what you will see are things that I said like yesterday look like stacks of coins. Okay? They're called the thylakoid. That's where that absorption of the solar energy and the electrons gaining energy happens. Yeah. Okay. So all of that goes on inside of there. Okay. Under an electron microscope, it looks kind of like this. Okay. Now, this is 1 micrometer across. So know that when we look at these, I say they're big. They're big, relatively speaking. Okay. Inside of a cell, having something that is, between 3 and 5 micrometers is pretty big. All right. Other organelles that we need to talk about. Okay, just double check in your notes that all of these are listed there. Okay, so the cell membrane, okay, because we haven't talked about that. The cell membrane controls these processes. So if this is what I would expect for you to write, okay, for the cell membrane. The cell membrane controls endocytosis and exocytosis. That is what I would expect you to write for the cell membrane, okay? Because that is the technical function of the cell membrane. Okay? I wouldn't want you to write it protects the cell. It doesn't actually do that, okay? It fails miserably at doing that. It controls endocytosis and exocytosis. That's movement into endo, okay, and out of exo, the cell. Now, the cell membrane does not look like how you probably expect. Okay? We get this idea that the cell membrane is like this line that just goes around the outside of the cell and it's featureless. But it's actually not. It's actually very complex. Okay? So the analogy I use is this. People picture the cell membrane to look like a thin layer of Jell-O. But the cell membrane is actually more like Jell-O salad. Okay, the Jell-O that's got fruit and other things embedded inside of it. Okay, so if you're really looking at what the cell membrane looks like, it actually looks like this. So you've got your Jell-O, that would be the double layer of fats, okay, so here and here, okay, but embedded all throughout that membrane are other materials, okay? Proteins, lipids, okay, glycoproteins, all kinds of stuff, receptor molecules, they're all covering the whole surface of the cell, okay? And a lot of those receptors are important for triggering certain things to happen within the cell, okay? So receptors are like antennas. They receive chemical signals from the organism that they're part of. So there would be a receptor on every cell for adrenaline. There would be a receptor on every cell for insulin. There would be a receptor on every cell for basically every hormone, testosterone, estrogen, whatever. Okay, there's going to be receptors all over the membrane for every one of those. Okay, when one of those is triggered, then they'll envelop the chemical signal, the hormone, and then it will do its job inside the cell. Okay, but it's just covered with stuff. It's not this layer that's featureless like we see in all of the cell diagrams, okay? It actually is a complex mix of a whole bunch of things, okay? So, on a quiz or a test, cell membrane controls endocytosis and exocytosis, okay? All right, cell wall. Now, we're talking about the plant's cell wall here. There are different kinds of cell walls. Fungi actually also have cell walls, but they're made of different materials. Okay? In a plant cell, the cell wall is not a wall. We talked about this yesterday, right? It's not a wall like that wall. Okay? It's actually a frame. Okay? So it serves no protective function whatsoever, because I get that all the time. People are like, cell wall, function of the cell wall, protect the cell. It does not. Okay? The cell wall is like a house that's just been framed. The walls will hold up the house, but they're no security whatsoever. You can walk through all of the walls because there's no drywall or sheeting on them yet. Okay? So the cell wall is actually a frame, and it's made out of cellulose. Okay? That's plant fiber. Okay? Now, is fiber important in your diet? Right? You get that from plant foods, right? Like beans and lettuce and things like that, right? Okay? Why is it important?
I don't have the answer, but what was the frame made out of again?
Cellulose. Right here, this word right here. Cellulose fiber, frame of the cell. Okay, so the purpose of the cell wall is it keeps the shape and supports the cell. We were talking about how yesterday that plant looks healthy because the cell wall is being supported from inside by the water vacuole. That plant does not because the water vacuole is empty and the cell walls aren't strong enough on their own. Okay, so back to the fiber question. Okay, fiber, okay, cellulose fiber, is actually not digestible by us. Okay? Herbivores can digest it because obviously all they eat is plants. We don't have an enzyme that can digest cellulose fiber, which means anytime we eat cellulose fiber, it goes right through us, unaltered. Think corn. Okay, if you eat corn, okay, think about it. You can't digest it. You can digest parts of it, okay? But, you know, you see the corn again. Gross, yes, but true. Okay, the outside of that corn kernel is made of cellulose, and you don't have any digestive enzymes that can break it down. So why is it so important for us to eat if I don't get any nutritional value out of it? Any of your parents eat Metamucil? Okay, mix the orange powder every morning. When you get old, you'll have to too. Okay. The purpose of fiber is that it helps to maintain the digestive health of your intestines. It's a brush. It helps clean you out. You don't want fecal matter, poop, to get stuck in places inside your intestine. It's not healthy because, well, it's gross. And you want it out of you. You don't want it to get hung up in places. Fiber helps to make sure that all of that stuff moves through properly. If you don't eat a lot of fiber, then you might struggle in the bathroom. There you go. Fiber is important. We can't digest it, we don't get any nutritional value out of it, but it's important for our digestive health. Okay, so here's what the cell wall actually looks like under an electron microscope. Okay, this is the cell membrane in purple. The green part is the cell wall. You can see it's hollow. Okay, it looks kind of like a framed house. You can see the walls, but there's no barrier. You can walk right through them. They'll still support the house, but they're not going to be any security. Okay, so it's a frame on the outside of the cell that tries to hold the cell's shape in place. Okay, and it gets helped by the water vacuole. The water vacuole inside is full of water, pushes everything against the frame, making it stronger. Okay? All right, so the water vacuole is the next thing, and it uses turgor pressure to support the cell from within. So cell wall and water vacuole have the same job. They support the shape of the cell. They do it in different ways. One is a frame from the outside, one using pressure from the inside. So this term, turgor pressure, that would need to be part of your answer. Okay? Turgor pressure to support the cell from within. Okay? And looking at this plant over here, you can kind of see what a big role turgor pressure plays in holding the shape of the leaves. Okay? I'll water this plant today, and then over the next few days you'll see it just perforate up. Okay? Those water vacuoles will fill back up again, and it'll look just like all the other plants. Okay, centrioles. Only going to be in animals, okay? So they're only in animals, okay? So structures in animal cells that aid with cell division. That's, I mean, what you really need to have if you were answering that question. Centrioles help with cell division, okay? But I was trying to explain what that looked like yesterday, and I drew a really crappy diagram. Then I found a better one, okay? So I'll let you write that down, and then we'll talk about the diagram here. Okay, so how this works is inside the cell are centrioles, and they're always paired together. Okay, so they're kind of hooked together. Right before the cell is about to divide, it'll actually duplicate all of its DNA, and it'll duplicate its centrioles. Okay, so you can see here that these are the centrioles, then they get duplicated, so you can see that there's kind of another one growing on each one. Then they separate, and they go to opposite ends of the cell. right? And when they do that, they send out these little fibers that will grab onto the chromosomes and then pull them to the correct side of the cell. Okay? Making sure that after cell division, no matter what happens to the shape of the cell, both new daughter cells end up with all of the DNA that they require. Okay? Everybody all right with that? All right, last one. Microfibers and microfilaments. Okay, the other name for that, we talked about this yesterday, the cytoskeleton. Okay, again, really only important in animal cells. They're kind of those bungee cords that keep the cell from tearing. Okay? All right, I'm going to give you a 3-minute break here before we start talking about transport. Okay, because I know it's just a lot of me talking, a lot of you listening, and I'm sorry about that. Okay, so just to kind of let you know here how the rest of the week is going to go. Tomorrow we're going to talk about all of the transport. We're going to start it today, but we'll finish it tomorrow. And then we'll do the pre-lab stuff for our lab on Monday. Okay, so we'll do the cell anatomy lab pre-lab stuff tomorrow. They will have the Chromebooks and we'll get that done so that on Monday morning we can go straight over to the lab and get looking at the microscopes. Friday, you have your quiz. Okay, that is on cell theory. Okay, and it is on modes of nutrition and use of the microscope. Okay, so none of the stuff we covered today, none of the stuff we cover tomorrow is on Friday's quiz. Okay, practice quizzes are 1 and 2, so make sure you check those out. Okay, it's going to be mostly recall stuff, fill in the blank, okay, that kind of thing. But just make sure you go over those notes thoroughly for Friday. Okay. And
For the microscope part, it's not like labeling. Like, what are we doing for the microscope? Like, what should I say?
like, I might ask you some stuff about how you use a microscope, maybe about estimating the size of an object. So I show you a picture, right?
Okay.
And I say, you're looking at this on high power where the field of view is 400 micrometers. How big is one of these things?
Okay.
So something like that. Yeah? Okay. All right. So the most important cell transport method is diffusion. Okay, we're going to talk about what I call the big 3. That's diffusion, osmosis, and active transport. Okay? But diffusion is the process that cells use within the cell. Okay, so through the cytoplasm, that fluid that fills up the cell. Now, diffusion is a natural process. It happens no matter what. Okay, it's not actually something the cell does in particular. It's just something that naturally happens. Okay, since you've all had this experience, I'm going to use it because you'll never forget it. Okay, but it works like a fart. Okay, if somebody in here farts, okay, eventually we'll all know, even if it's a silent but deadly one. Okay, we'll still all eventually know because the smell will diffuse through the air from a point of very high concentration to areas of lower concentration. Okay, everything moves down the concentration gradient by diffusion, whether it's through the air or through a liquid or anything like that. Okay, if you've ever put food coloring in water, you can actually watch the process of diffusion happen. You can go home and try it. Okay, you put that drop of food coloring in the water and it eventually kind of spreads out everywhere. You can see it moving in all directions, from high concentration to low concentration. Okay, diffusion always moves material from high concentration to low concentration. So generally, it gets material where it's needed. It's needed where it's in low concentration. And it's naturally going to move from where it's being produced in high concentration. All right, and it'll naturally just diffuse down the gradient. So you can see that in this picture right here, where they're injecting dye into water. Okay, here's the area of high concentration, but it's moving away and becoming less and less concentrated, okay, as it moves down the gradient. Okay? So the rule for diffusion is this, okay? In the absence of other forces, a substance will diffuse from where it is more concentrated to where it is less concentrated. We call that down the concentration gradient. Okay? That process is passive. Okay? It requires no energy. It just happens. Now, because it just happens, it's also not something the cell can stop. Okay? Diffusion is just a natural process. The cell just happens to use it because it's convenient and free. Okay? But diffusion is kind of like free shipping. Okay? You get what you pay for. Right? It's going to be slower. Okay? If you pay for shipping, it's usually faster. Okay? But the free shipping is slower. You don't get your package nearly as quick. Okay? The other problem with diffusion is that it's not targeted. Okay? It's not, I have addressed this molecule to go there. It's just going to go everywhere. Okay? It's going to go from where it's concentrated to where it's less concentrated, which means it'll probably end up where it's needed. Probably. Okay? Is it a guarantee? Absolutely not. Okay? So, again, you get what you pay for. The cell doesn't have to expend any energy to use diffusion, but it also isn't guaranteeing that what it made got where it needed to go. And it probably does. The other issue with diffusion is because it's just spreading out, okay, does it get pretty diluted pretty quickly? Okay, so if I'm looking at a cell, and remember, a cell is small. Okay, so if I've got this cell and the substance is being produced right here. Let's just say that every one of these dots represents the substance that's being produced. Okay, that's where it's in high concentration. Everywhere else, it's going to be lower in concentration. So these things are naturally going to kind of move away from that point of production. Okay, by the time they get this far away, they're further apart. Okay, well, there's just as many dots over here, but they're pretty far apart. Okay, if the target was right here, only maybe one of those dots actually ends up there. Right? So diffusion's fine if you don't need to guarantee it gets where it's going, and you don't care how fast it happens. Okay? And the cell generally doesn't. Okay? Because a cell is small. Over the size of a cell, can this spreading out that I've kind of greatly exaggerated here, does it happen that much? Not really. As long as a cell's not too big, diffusion works great. Okay? Because it doesn't get a chance to get that spread out. That's why, earlier today, I said there's kind of an upper limit to the size a cell can reach. If a cell gets too big, then diffusion doesn't work at all, because by the time stuff gets to the other side of the cell, it's so diluted that it's not getting where it needs to in sufficient concentration. Okay? That's actually what limits the upper size of a cell, is the cell's dependence on diffusion to transport material. Okay? All right, so what do we need to know about diffusion? Okay, it's free, so passive. Okay? Diffusion is passive. It moves down the gradient, and it happens within the cell. So it moves through the cytoplasm. All right. We're going to leave it there for today. We're going to end a little bit early because I've just been doing nothing but yak at you all day. Okay?